NAD.
Precursors like nicotinamide mononucleotide, NMN, and nicotinamide riboside, NR, are gaining widespread attention in the longevity research community for their potential to restore cellular energy, enhance metabolic resilience, and protect against the molecular damage that accelerates aging. At the heart of this enthusiasm lies NAD, nicotinamide adenine dinucleotide, a coenzyme that plays a central role in redox reactions and serves as a critical signaling molecule in pathways that govern cellular stress responses, DNA repair, and mitochondrial function.
Importantly, NAD
Levels decline significantly with age, contributing to reduced energy production, increased oxidative stress, and impaired genomic maintenance, including at the level of telomeres.
Boosting NAD.
Availability through precursors like NMN and NR, researchers hope to reinforce the cell's natural repair mechanisms, including those that preserve telomere length and stability. One of the most promising mechanisms through which NAD supports telomere integrity involves the activation of sirtuins, a family of NAD-dependent enzymes that regulate key processes related to aging and stress resistance. In particular, SIRT1 has been shown to play a role in maintaining telomere structure and promoting the stability of the shelterin complex, a protein assembly that protects telomeres from being mistakenly identified as broken DNA. SIRT1 also supports the function of telomerase, the enzyme responsible for adding telomeric repeats to chromosome ends, especially in stem cells and immune cells.
Facilitating sirtuin activation, NAD.
Precursors may thus offer an indirect but potent means of reinforcing telomere maintenance. Another key benefit of NAD restoration lies in its support for DNA repair processes. Cells are constantly exposed to genotoxic stressors, oxidative radicals, UV radiation, and replicative errors that can damage DNA, including telomeric regions. One of the most important enzymes in this context is PRP, poly-ADP-ribose polymerase, which uses NAD as a substrate to initiate repair of single-strand DNA breaks. Without sufficient NAD, PRP activity diminishes, leading to accumulation of DNA damage that can destabilize telomeres and accelerate their shortening. Supplementing with NAD, precursors helps maintain the pool of NAD needed for effective repair, reducing the likelihood of telomere attrition due to unrepaired DNA lesions. Mitochondrial health is another major link between NAD and telomere preservation. Mitochondria are both a source and a target of oxidative stress, and their dysfunction is closely tied to aging and telomere instability.
NAD
Is essential for mitochondrial function and biogenesis, particularly through the PGC-1A pathway and sirtuin activation. By rejuvenating mitochondrial performance, NAD precursors may reduce the production of reactive oxygen species that damage telomeric DNA and surrounding nuclear structures. In essence, a cell with high NAD levels operates more efficiently, produces less oxidative stress, and is better equipped to defend its genomic ends from erosion. Human trials of NMN and NR supplementation are still in their early stages, but have so far shown promising results in terms of safety, metabolic health, and markers of cellular vitality. Studies have reported improvements in insulin sensitivity, vascular function, and muscle endurance in middle-aged and older adults, while direct evidence of telomere lengthening in response to NAD boosters is limited. Several trials have noted reductions in inflammatory markers and oxidative stress, two of the key drivers of telomere shortening. These systemic improvements suggest a favorable environment for telomere maintenance over the long term. Another advantage of NAD precursors is their favorable safety profile and ease of administration. Both NR and NMN are orally bioavailable and well-tolerated at doses up to several hundred milligrams per day. NR has already achieved GRA, generally recognized as safe, status by the FDA, and NMN, though less commercially developed, is being actively studied in both academic and pharmaceutical contexts. The accessibility of these compounds makes them attractive candidates for preventive strategies aimed at healthy aging. Future research will likely clarify whether NAD supplementation can directly influence telomere length in measurable ways, especially when combined with other interventions like exercise, fasting, or sirtuin-activating polyphenol. There is also growing interest in understanding how different populations, such as older adults, individuals with chronic disease, or those with accelerated biological aging, might respond to NAD. Restoration strategies. Personalized approaches could maximize benefits for telomere preservation while avoiding potential downsides of overactivating certain cellular pathways. In conclusion, while NAD precursors like NMN and NR do not lengthen telomeres directly, they support a constellation of cellular functions: sirtuin activation, mitochondrial health, DNA repair, and inflammation reduction that collectively protect against telomere erosion. Their ability to bolster the underlying infrastructure of the aging cell makes them one of the most promising and versatile tools in the longevity toolkit. As ongoing clinical studies expand our understanding, NAD enhancement may soon become a foundational component of strategies designed to preserve not just telomeres, but the full spectrum of cellular youthfulness and vitality.